Okay, so hi everyone. My name is Jessica, and I'm here presenting work done on behalf of Dr. Tao Lu at UVic. This research introduces a biologically inspired architecture called BNN, which stands for Ray Trace Biological Neural Networks. So as Steve mentioned, my name is Jessica Sevick. I'm a research partnerships officer at UVic. I have an undergraduate degree in civil biomedical engineering, not a lot of overlap there. I did my master's in experimental medicine at UBC, and I'm actually, fun fact, a two-time Olympian in the sport of rowing. But today I'm here to highlight the work of Dr. Tao Lu. Dr. Lu leads the Bio and Nanoinformatics Research Lab at UVic, which focuses on quantum integrated bio and nanophotonics, machine learning, and spectroscopy analysis. His interdisciplinary approach bridges the fundamentals between physics and computer science with practical applications in technology. Full disclosure, I am not a computer scientist, so bear with me on the technical sides of things, but I'm going to do my best. So as I mentioned, this work introduces a biologically inspired architecture called BNN. Instead of viewing neural networks as flat layered graphs, BNN reimagines them as 3D biological systems, ones that develop, connect, and adapt much like biological tissue. We'll take a look at why this idea was developed. Here's a quick little video. How it works and what the experiments show, and where its implications and what they are for future AI research. This is, as Todd said, he's much more on the applied science side, whereas this is much more theoretical, so hopefully you find it interesting. So what are some of the current limitations of standard ANNs and why Dr. Lu's lab was looking at the biological side? The first is that classical ANNs have fixed topology. The number of layers in neurons is predetermined before training. Once the network is built, the architecture itself cannot change. This rigidity prevents it from adapting to new tasks or input features without having to retrain it from scratch. Standard ANNs typically only adapt weights, not connectivity, and this leads to limited transfer learning. As most of the transfer learning addresses weights only, it cannot expand to accommodate new features. Yay. There is also no built-in 3D spatial or 3D structure with the classical ANNs. Connections are abstract. Neurons don't have any spatial positioning, and connectivity doesn't consider geometry or distance. To summarize, while these classical ANNs are powerful for static, well-defined tasks, their rigidity in structure and inability to dynamically change limits their ability to do transfer learning. BNN directly addresses these limitations by combining 3D placement, adaptive connectivity, and structural plasticity. So why did they look at biology? It's because biological brains are by far more efficient and adaptive than, in general, than artificial ones. They can rewire, they can learn new tasks, they can recover from injury. These principles suggest that spatial organization, structural flexibility, local connectivity, and heterogeneous activations could make artificial networks much more adaptable, scalable, and efficient. So to understand the inspiration behind BNN, we're just going to do a brief history of, or brief refresh on neuroanatomy. So neurons are the core computational units of the brain and are controlled by ion channels, and these transmit electrical signals. Dendrites are what receive the signals, and axons are what send them out. Glial cells are more supportive units for the brain, and they regulate and block connections. And something called synaptic plasticity refers to the ability of the connections to strengthen and weaken over time based on activity. And finally, biological networks are organized structurally in modularity. Clusters of neurons often correspond to one functional area, with the connectivity being distance dependent. As we'll see in the following slides, our BNN combines all of these principles to create a more spatially grounded, biologically plausible network architecture. So, as I said, BNN is designed as a 3D spatial network where the neurons, he calls them neurons, are positioned in a 3D sphere. The inputs are mapped onto the surface to preserve the local topology. There's hidden neurons randomly distributed within the sphere, and the outputs are clustered near the center. This spatial organization represents what an actual brain would kind of look like. Non-neuronal glial cells are obstacles that regulate the connectivity. The connection can only be made between the neurons in a straight line with a ray or ray tracing algorithm, and this only happens if there's no glial cells blocking that connection. The adjacency matrix that is used is sparse, and this is also reflective of the biological tissue, and the architecture is also dynamic. Neurons and connections can be either pruned and removed or added if they needed for new features. Each neuron also has a learnable parameterized activation function that can evolve during training and allows heterogeneous firing behaviors, which in itself lends itself to the oops, I went back, my bad. Sorry, I really skipped ahead here. 1 second, technical difficulties. So basically, our BNN is not just a neural network, it's a biologically inspired 3D computational system where structure, geometry, and connectivity are part of the learning process. So to make our BNN work, they needed to wait to determine which neurons can actually connect, essentially through which ones can see each other through the ray tracing technology. This was handled through a series of ray tracing algorithms. RT1, the first, is the simplest approach where every neuron checks its visibility with every other neuron. This system is very accurate, but scales quadratically, so is not very efficient and not applicable for anything other than small systems. RT2, or ray tracing algorithm 2, improves this by introducing probabilistic pruning. Neurons that are far apart may be skipped or sampled at random. This introduces stochastic variability, which is representative of biological tissue. However, it also led to instability in the resulting networks where some neurons were either isolated or underconnected. And then the preferred algorithm, or RT3, limits each neuron's ability to check only within a specified radius or specified distance to the other neurons. This brings the complexity down to scale linearly based on the average number of local neighbors. And using RT3, we can simulate networks with around 240,000 neurons and 240,000 glial cells and fully resolve all of their connections within 20 seconds. The RT3 kind of finds the sweet spot between RT1 and RT2 with realistic local connectivity, sparse, but with a sparse matrix and plausible wiring, making it computationally practical. In traditional deep networks, we choose a fixed activation function, typically ReLU, TANH, sigmoid, or GELU. And every neuron in the layer uses the same nonlinearity. Oh my God, help me. While that works well computationally, it is far from the biological reality. In biological neurons, each cell type exhibits a unique input-output response curve shaped by its ion channels, its membrane properties, and synaptic environment. Some neurons are sharply thresholded, and while others saturate gradually, and some are biphasic or even oscillatory. This diversity is central to the efficiency and the stability of computation in the brain. That's why Dr. Lu introduced a universal activation function, or UAF, that brings this heterogeneity into the array BNN. Mathematically, as you can see, each neuron has its own activation function where the function is drawn from a parameterized family of functions and a set of learnable parameters. The parameterization can combine several standard forms. For instance, during training, the parameters A, B, C, D, and E evolve along with the weights, meaning that each neuron learns not how to just respond, but how strongly to respond. This allows the network to self-select the best linear or non-linear behavior based on each layer. It enables functional specialization. Some neurons become more linear for stable signal propagation, while others are highly non-linear for feature selection. It also smooths gradient flows because the network can morph into non-linearities during training, mitigating vanishing and exploding gradients. And during transfer learning, the UAF parameters adapt rapidly, allowing the network to retune its activation behavior without full retraining. So one of the most innovative and biologically inspired aspects of BNNN is its approach to transfer learning and adaptation, not only at the level of the weights, but at the level of structure. In conventional neural networks, when you perform transfer learning, you typically freeze early layers and replace or fine-tune later layers and then retrain it. Sorry, I lost my place. The architecture itself, i.e. the number of layers, the number of neurons, and how they connect remains fixed. But in the brain, that's not how learning works. Biological systems exhibit structural plasticity, enabling the nervous system to adapt to new tasks and sensory modalities without overwriting existing representation. So our BNN brings in this principle into the computational domain. Mechanistically, when new input features appear, the model adds new input neurons onto the surface of the 3D sphere. Ray tracing is then used to recompute the connectivity locally, and nearby hidden neurons either form new rays if glial skills allow that to happen. This preserves the spatial modularity and limits disruption to the existing structure. During the fine-tuning, low activity and low weight connections are pruned, freeing up capacity and improving generalization. This allows the BNN to perform architectural level transfer learning. Instead of just transferring the weights, it transfers the topology itself. And the sparse adjacency matrix is updated dynamically, adding and removing neurons as needed. Importantly, previously learned neurons are preserved, preventing catastrophic forgetting. And from a learning theoretic perspective, this introduces a structural a priori. Nearby neurons that have been co-activated in the past are more likely to remain connected, guiding adaptation in a biologically plausible way. So Dr. Lu's lab tested BNN on several domains to evaluate whether this was actually going to yield computational benefits. For the task, both regression and classification problems were used to cover continuous and discrete output behaviors. For baseline comparisons, BNN was compared to standard MLPs and CNNs of roughly equivalent parameter counts. For the data sets, they used the Alcala data set, which is an indoor localization data set, and the EEG motor imagery data set, which classifies human EEG signals. These tests were stress tested the model's ability to adapt to new inputs and noise. I'm losing the ability to speak. So as mentioned, the Alcala data set is an indoor localization task. Essentially, establishes a device's position from a set of wireless signal strengths. The data set is challenging because it's high dimensional, it's extremely noisy and often incomplete, making it a good test for structural adaptability. Despite the unconventional architecture, BNN achieved similar or slightly better accuracy, but the real difference was in its ability to be efficient and adapt. BNN reached convergence in fewer epochs, especially when the data was limited. This is likely due to the sparse connectivity and the UAF, which reduces the redundant computation and improves the gradient flow. When new signals were added, a traditional ANN needed reinitialization to handle the new input dimension. BNN, however, simply added new input neurons on the sphere's surface, extended the local connectivity through the ray tracing. The model adapted and expanded and performed the same task in roughly 20 to 30% faster in the low data regimes. This highlights its suitability for environments where data sources evolve over time, and which is really not applicable to static networks. The second test was the EEG motor imagery dataset, which is a task where the model must classify EEG signals associated with imagined hand movements. This is an excellent stress test for the BNN because EEG data is non-stationary, has low SNR, and is highly subject dependent. Traditional deep models often struggle with generalization and catastrophic forgetting when transferring across subjects. BNN achieved the classification accuracy on par with the CNN-based baselines, but was able to adapt quicker to the inter-subject variability. So let's move from task-specific results to characterization of BNN as a computational system. A critical question was scalability. Could a biologically inspired 3D network actually scale efficiently? The core ray tracing operation implanted in the RT3 engine scales linearly with the number of neurons, as I said, up to about 240,000. The nonlinearity is non-trivial because in naive implementations, ray tracing grows quadratically with object count. The optimization levels spatially partitioning and boundary volume hierarchies similar to those used in computer graphics rendering. The resulting sparse adjacency matrix allowed to have a small number of bound constants. And when you visualize it, the topology strikingly looks like the cortexes of the brain, where it shapes and flows, creating around that sphere. These analyses suggest that Ray BNN is not only a biologically inspired in concept, but also an emergent structure. It forms spatially modular, efficient architectures that scale gracefully, and an encouraging sign for future biologically grounded AI systems. So obviously, with any ambitious new architecture, it has limitations, both conceptual and practical. Although the scaling is nonlinear, ray tracing remains more computationally expensive per neuron than simple matrix multiplication in ANNs. This makes it less suitable, This makes it less suited for extremely large-scale tasks like natural language modeling, where billions of neurons would be infeasible without hardware acceleration. Also, the glial cell's density strongly influences the connectivity. Too few glia and the network becomes overly dense, and too many and it fragments. Optical density depends on the task dimensionality and noise level. And then obviously, imagining something in a 3D space is quite complicated and requires validation as well. So limitations aside, what does Ray BNN mean for the broader field of AI? First, it challenges one of the general assumptions of deep learning, and that is that the architecture is fixed. Ray BNN treats architecture itself as learnable, adaptive component of the model. This opens a new paradigm, the meta-learning of the structure. Instead of searching for the best architecture offline, as in neural architecture search, the model can evolve on its own structure online as part of the learning. Second, Ray BNN introduces spatial geometry into a neural network. Traditional ANNs operate in abstract weight space. Ray BNN grounds neurons in physical metric space. This spatialization enables distance-dependent regularization and potentially more interpretable topologies. Third, the ability to grow, prune, and adapt connections dynamically makes Ray BNN a natural candidate for continual learning systems. Models can accumulate knowledge over time without catastrophic forgetting. And finally, it exhibits a broader movement in AI, which is bridging biological and artificial intelligence. By encoding biological constraints like glial mediation, sparse connectivity, and neuron heterogeneity, we move towards systems that are not just more efficient, but also more robust and flexible, with properties evolution has already confirmed. So future directions, there are several new exciting directions for Ray BNN, both in computation and potentially even in biology. First is the integration with graph neural networks. Conceptually, Ray BNN is spatially grounded, self-organizing graph. Coupling it with GNN messaging passing systems could yield networks that learn both what to communicate and where connections should exist in space. This might form the basis for hybrid architectures, geometric GNNs whose structure evolves through biologically inspired ray trace growth. Second is hardware acceleration. Ray tracing has matured enormously in computer graphics, and we now leverage dedicated hardware like NVIDIA's RT cores. Implementing Ray BNN directly on such accelerators can make large-scale dynamic 3D networks feasible in real time, opening possibilities for interactive simulations and structural learning. Third is real-time structural plasticity. The current Ray BNN adapts structure between training phases. The next step is online adaptation, neurons and glial proxies adjusting continuously as new data arrives. This would enable lifelong learning in a system that never stops training. And fourth is theoretical formalization, developing a mathematical framework, and describes convergence, stability, representational capacity in a dynamic, self-restructuring networks is going to be crucial. And fourth, and finally, RayBNN lends itself for the expansion with potential biohybrid neural interfaces, which leads us to Dr. Lu's current work, which is training living neurons to emulate the behavior of the artificial neural network. He's literally growing neurons in his lab. As a computer scientist, is growing neurons in his lab right now. But this slide describes basically the experimental framework that's going to bridge between the RayBNN learning and the neurobiology. This is done first by genetically modifying the neurons using optogenetically transfected virus vectors. So basically, they take the part of the virus that is sensitive to light and they introduce that into genes to grow the neurons. A biosensor virus is also introduced, enabling the neurons to emit fluorescent signals when they're activated to see if it works. This is followed by encoding the training data that features the vectors and model weights from RayBNN into optical signals, and using a pixel array like a digital micro mirror device or a micro LED device, the project projects the patterns of light onto the neurons. When illuminated, the neurons process the optical input through their own electrophysiological networks, and their responses, which is their ion influxes, their action potentials, and their calcium releases, are captured through fluorescence microscopy. These biological responses are then digitized and compared to target outputs generated by the reference RayBNN, and the computing system then evaluates the difference between the biological and the digital outputs. It then adjusts the optical stimulation parameters to reduce its error, and this constitutes a closed-loop training cycle, analogous to backpropagation, but is implemented via light modulation and cellular plasticity, not gradient descents in code. Over many iterations, the living neuronal culture learns to reproduce the output behavior of the RayBNN, and the result is a trained biological neural network capable of performing computational tasks. So here's a slide. I'm showing what's going on in his lab right now. So they built the microscope and the setup, and as you can see, the top image is just a commercial microscope, but then he also built his own to capture it, but the neurons are currently responding to the RayBNN network, which is super exciting. And to quote Dr. Lu, that he thinks biological hosts are much more efficient than silicone hosts. So thank you for bearing with me through that. This is not my area of expertise. This highlights what RayBNN is and what it can do. This is other AI. I'm a research partnerships officer, so basically I'm just trying to connect people in the industry with people at UVic to either get their research out into the real world or help companies partner with UVic, because you can leverage funding, do specific research projects, and those are some of our other projects that are going on. And thank you.